Hey everybody, Mr. Power here. Welcome back to the lab. Today we're going to be looking at your photosynthesis required practical for AQA, both triple and combined science. Okay, just as a recap of photosynthesis, our reactants for photosynthesis are carbon dioxide and water. So a plant will combine carbon dioxide and water with the use of light. Now we write light above because light is not one of our reactants or products. It just provides the energy for the reaction to take place. So carbon dioxide and water gives us glucose and oxygen. Now, dealing with looking at our products and trying to measure the rate of photosynthesis, it can be quite difficult, especially because glucose is very hard to see, and oxygen in the air, if we're dealing with a land plant, is also difficult to see it expel. So what we do is we use Elodea pondweed, and basically this is an aquatic plant that we can very easily see the photosynthetic rate by looking at the oxygen being released. Now, the setup of this practical is quite simple. We're going to use light intensity, so we're going to use a source of light, which is a lamp. We're going to be looking at Elodea, and then we have our meter stick. And what we're going to be doing is we're going to check to see what the effect of different light intensities have on the rate of photosynthesis. Now, with any experiment, it's really important that you identify your variables. So we have our independent variable, which is the thing that we change, our dependent variable, which is the thing that we measure, and our control variable, which is the thing that we keep the same. So in this experiment, the thing that we're changing is the distance, or the light intensity. We're going to be measuring the number of bubbles that are being produced. That's our oxygen, and the control variable is going to be temperature. So our first trial is looking at a distance of 10 centimeters between the source of light and our Elodea. And we can see that when the light is close and the light intensity is very high, there is a lot of oxygen being released. Okay? Typically, we would count these per minute, and that would show us how much oxygen was being produced or how many bubbles of oxygen were being produced per minute. We then move our Elodea 10 centimeters back, and that will make the light intensity less, and let's see our result. And we can see as we've moved farther back, the light intensity has gotten less, and we can see the rate of oxygen production has gone down slightly. Still quite high because the light intensity is high, but it has gone down slightly. Now we moved our Elodea far back, low light intensity. Let's see our results. And we can see that the number of bubbles being reduced has gone down yet again. So for this experiment, what we've learned is that as you increase light intensity, you increase the number of bubbles that get released. So as you increase light intensity, you also increase the rate of photosynthesis. Now, this will only happen up to a certain point because then we get into what are called limiting factors. And one of the things you have to be careful of in this experiment is to make sure you add something called sodium hydrogen carbonate. Now that is going to supply the carbon dioxide for our plant. So that means that they can use the carbon dioxide from the sodium hydrogen carbonate and the water to perform.